Hi guys, welcome back to my channel. I'm so glad to see you all here. So in this video, we will be talking about a platform that makes dataset discovery and retrieval effortless. So in this video, we will be exploring the power of bulk OmixAtlases, which is a revolutionary technology that enables researchers to analyze gene expression across various cell types and tissues. So this video is in collaboration with Elucidata, which is a data science company that specializes in developing software and data analysis solutions for the life science industry. Poly is their AI-enabled cloud platform, which is a one-stop platform to search and explore curated biomedical data. So Poly's OmixAtlases provides access to large number of curated bulk RNA sequencing studies. So whether you are a seasoned researcher or you're just starting out in this field, this video is packed with insights and tips on how to take advantage of this platform, of this bulk OmixAtlas platform, and achieve breakthrough results in your gene expression studies. So without further ado, let's get started. So before going into the details of Poly and the OmixAtlases, let us talk about some of the public sources of gene expression data where most of us extract the RNA sequencing or the bulk RNA sequencing data from. And the first and the most commonly used source is the Gene Expression Omnibus by NCBI, which is a repository for gene expression data, including the RNA sequencing data, and it has a collection of data sets from a wide range of studies and organisms. We also have Sequence Read Archive, which is a repository for the high throughput sequencing data, which also includes RNA sequencing data and is maintained by NCBI as well. Next, we have ArrayExpress, and ArrayExpress is a repository for gene expression data which contains RNA sequencing data as well and is maintained by European Bioinformatics Institute. Next, we have the TCGA or the GDC portal, which is the Genomics Data Commons by NCI. And this is the portal where we extracted the data from in our previous video using a package called TCGA BioLinks package. And this portal is specific to cancers and it has data from a wide variety of cancers and histologies. The European Nucleotide Archive is a repository for raw and processed sequence data, which includes the RNA sequencing data and is maintained by the European Bioinformatics Institute as well. And lastly, we have the Human Transcriptome Atlas, which is a resource that provides a comprehensive characterization of human transcriptome using bulk RNA sequencing. So these are some of the very common and popular repositories, publicly available repositories where the RNA sequencing data are deposited and one can download the RNA sequencing data from each of these sources. However, there are certain challenges that are associated with downloading data from these public sources that I will be talking about in a little more detail in the next slide. So talking about specific challenges associated with searching datasets on these public repositories, and here talking mainly about NCBI's GEO, as this is one of the most widely used repositories while one is looking for an RNA sequencing dataset. So there are some of the challenges that are associated with looking up datasets on NCBI GEO. So the first and the foremost is that GEO is large and complex repository, and there may be inconsistencies in the way data is reported, such as in the use of different sample annotations, platform specific issues, and even missing data. So apart from data inconsistency, another challenge of looking up dataset is that RNA sequencing datasets can be generated using different protocols and platforms, which leads to differences in the data that can make comparison and analysis difficult. In addition to that, RNA sequencing data can be generated from a wide range of organisms and tissues, and the data may have different characteristics such as read length, difference in the depth of sequencing, and expression levels, making it difficult to compare datasets from different studies. In addition to data heterogeneity, another challenge is that RNA sequencing data can have complex data structures, such as there could be multiple samples per study, technical replicates, and batch effects that can also make it difficult to extract meaningful information from the data. And the last and the most important limitation is that GEO provides a search engine for users to search for datasets, but the search capabilities may be limited and not always be intuitive to use, so making it difficult to find the specific datasets of interest. Also, the use of logical operators using keywords yields limited results. In addition to these challenges of looking up datasets on NCBI GEO, there are also challenges associated with data that are present in GEO. Even if we do manage to find the relevant datasets of our interest from GEO, there are still challenges that we face with the data that we download from GEO. issues like data quality and consistency. So GEO data is generated by many different laboratories using different methods and platforms, which can result in variable data qualities and inconsistencies among the data. In addition to that, we face the issue of data annotation with the GEO data, as GEO data is poorly annotated, making it difficult to interpret and compare results across different studies. GEO data can also be difficult to work with due to the various file formats and data structures used, making it time consuming to extract and analyze the data. It also makes the automation with these data a little difficult, because since the raw data is saved in different formats, it becomes the automation of extracting the raw data and processing challenging. In addition to that, we face platform specific bias with this data, as different gene expression platforms such as microarrays and RNA-Seq can produce different results, which can result in inconsistencies when comparing data from different platforms. Variation in sample preparation methods can result in inconsistencies in gene expression data. In addition to sample preparation, normalization is an important step in gene expression data, and different normalization methods can result in different interpretation of the results. Apart from the normalization, the quality of the data submitted to GEO can vary, with some studies having low quality or incomplete data. And different data processing pipelines and algorithms can result in different results or different file outputs, particularly for more complex data types such as RNA sequencing. So in different data processing methods can also be a challenge when we are trying to extract data from different studies and trying to compare the results from these different studies. And lastly, technical replication is important for gene expression analysis, but the level of replication can vary between the studies. So introducing OmixAtlases on the Elucidata's Poly cloud platform. So basically, OmixAtlases aims to address these issues that we spoke about in the last two slides at two levels. It addresses the issues that are faced in the data, and it also provides solutions to look for the relevant data sets. So talking about addressing the issues in the data, it ensures that the metadata from different data types and across different data sources have been curated and harmonized and made ready for downstream machine learning and analytical applications. It also provides a very intuitive graphical user interface platform where you can use point and click solutions to look for the relevant data sets. Some of the features of the OmixAtlases are that it provides access to 26 different data types from over multiple data sources. It's constantly evolving, so it is keeping up with the rapid pace of biological data generation, and it's updated frequently and is in sync with the source repositories. So whenever there are new data sets available on these public repositories, OmixAtlases are constantly updated with these new data sets. All the types of data from the diverse sources are structured. The metadata have been harmonized with controlled vocabularies and ontologies, and curated metadata makes data sets searchable and findable across data sources. And lastly, OmixAtlas also allows querying of the data through point-and-click solutions, as I previously mentioned, and code-based advanced access allows one to use the data on Poly on the platform, or can be and the data can be downloaded and can be used outside of the platform. The bulk OmixAtlas contains over 40,000 datasets, and this is like a huge amount of data that is available on a single platform, which encompass over 800 tissue types, over 2,600 diseases, so many organisms, so many cell lines, so many cell types. And a major source of this data are the data that is deposited on the NCBI GEO. So today I'm interested to check for expression of BRCA1 gene in triple negative breast neoplasms, and I want to fetch 3 publicly available bulk RNA sequencing datasets with study design involving data from triple negative neoplasms. And my query would look something like this, which is a fairly simple and straightforward query. And I will be using this query on both the OmixAtlas platform as well as on the NCBI GEO. And I sort of want to make a comparison as to how many and how relevant datasets I'm able to retrieve using this query on both the platforms. In addition to that, I also want to talk about additional features OmixAtlas provide in terms of filtering the dataset, finding the relevant data, as well as visualizing the cohort and the metadata associated with the cohort, which helps us determine which are the datasets which are relevant and of our interest, and which are the datasets we would eventually want to include in our studies. So in order to access the OmixAtlases on Elucidata's Poly's platform, one needs to create an account and sign in. And once when you sign in, you get to see this page where you can get access to various OmixAtlases. So currently there is a bulk OmixAtlas and a single cell OmixAtlas. And I'm interested in a bulk, in getting bulk RNA sequencing data. So I'm going to click on the bulk RNA-Seq OmixAtlas. And when you click on that, this sort of takes you to like an introductory overall information page where it shows you the amount of data that they have on their portal currently, which is a lot of data sets, and encompassing a lot of tissues and diseases and organisms. And you can also get the distribution of the number of data sets according to the diseases, the organisms, the current data type that they have, and the source of the data that they have the data sets from. And you can click on view all data to sort of go onto their page where essentially you can get this graphical user interface where you can look for data sets, you can search for data sets, there are filters available, and there are various data sets that are displayed right now. Before going into this detail, I would want to go back to the GEO dataset browser and use the query that I previously spoke about and see how many datasets I'm able to retrieve and how relevant are those datasets. So let us go to the GEO dataset browser, and this is the browser where you can put in the search terms and look for the relevant datasets. So here I'm going to put the search terms in my query. So I'm interested in the data from the breast cancer and triple negative neoplasms. And when I hit enter using the search terms, I get like these 4 data sets, out of which 3 are more relevant to me because I'm interested in data from Homo sapiens, not from the mice. And to further get information on each of these datasets, I can just click on them and I can get like an overview of the dataset record. And from the record, I can see that one of the dataset here is looking at the platform. This is a microarray data. This is not an RNA sequencing data. So again, this is not a relevant dataset that I would want to download and use in my analysis. Clicking on the other dataset, again looking at the platform, this is an microarray data. This is not an RNA sequencing data. So again, this would not be included in my analysis. So you can see that first and foremost that I don't have that many options when I use the search terms in terms of a list of data sets that I can choose from. I just have a limited options to choose from the data set. And again, I don't know very specific or exact terms to look for the relevant data set. So searching using keywords in the dataset browser by NCBI GEO is a little challenging to look for the relevant data sets. So now that I did not get any relevant or much of the relevant hits using the search terms or the query, let's go on to the Poly platform using the bulk OmixAtlas, and let us use the same query that I used on the NCBI GEO. So here I will be writing breast cancer and triple negative neoplasms. This is the exact same query that I used on the NCBI GEO. And when I hit search, I get so many of the matching datasets. And just to make sure that these are from the Homo sapiens, I will be using the filters that are provided on the left side of the page. And here I will click on the organism that is Homo sapien. Again, at the beginning, you will see that there are around 5,600 datasets which are retrieved based on my search terms. And this is far more relevant in terms of it is fetching me all the data sets that are breast cancer, as well as the data set that the study includes the triple negative neoplasms. And there are so many of the data sets which gives me more options to choose the relevant data set from. And it is also helpful that it shows you the query that you're building. So basically, these are the data sets that are matching your search terms, and this is the filter that you essentially used. And there are also additional filters that are available on the left side of the page, where you can select specific cell lines if you're interested in the data that are generated in specific cell lines, or you can also add additional diseases in addition to the breast cancer or additional neoplasms. You can also add the drugs that might have been tested in the study, which might be of your interest. So there are various other filters that you can apply and refine your search, which will really be helpful in terms of searching the relevant data set that you would want to essentially include in your studies. In addition to these useful features, there are also another feature that I really like about this portal, is that it provides you with the prompts for the keywords that you can use to make your search more refined. So let's say you just started with breast cancer and you did not know what else you would want to search and what other keywords that you can use. But once you have searched using some keywords, it can prompt you and help you to pick another keywords that might be of your interest in terms of the data set that you're looking for, and you can use these keywords to build a new or more specific query that can give you specific results. A couple of more features I want to talk about before we jump into looking at these data set. So while we are building a query, since I used a logical operator and to build my query, you can also use additional logical operators like or, not, or you can even group your query, or you can also use exact terms to search for your data sets. In addition to that, you can apply as many filters. This is something that I previously spoke about. You can use as many filters as you want. You can also restrict your searches for dataset which study a specific gene of your interest. In addition to that, once you have the relevant matching datasets, you can also sort the list according to the relevance, according to dataset ID, the number of samples, or how recent the dataset is. And if there is a dataset that you do not see in the list of retrieved dataset, you can also request that particular dataset. Now, having said that, let us take a look at the details or the metadata or the data that is associated with these dataset, which will help us determine if this is relevant for our analysis. So I'm going to click on the second dataset on the view details button. So for each of the dataset, you can click on view details and it will give you more information about the information that is present in that dataset. So I'm going to click on the second one, which has 6 samples. So I'm going to click on view details here. And when I click on that, I get like an overall view of this dataset in terms of what is the overall design of this dataset, what is the abstract that the dataset is associated with, from what paper it's associated with, the number of samples, also the source, the link to the source. And when I click on it, it will take me to the NCBI GEO record page for that dataset. And when I scroll down, I get to see some informative charts here, which tells me more about this dataset in terms of whether this dataset contains any control samples, what is the disease stage for each sample here, and what percentage of the samples are triple negative breast neoplasms, what percentage of the samples are just breast neoplasms. I can also get information on what is the cell line that is a part of this study, what are the type of the cells, and also whether there are any genetic modifications that are performed on these samples or the cell lines, and if there are any modifications, what are the type of modifications. And these are interactive, so you can just hover over these bar graph or these pie charts, and you can get more information on what these pie charts are depicting. And when I scroll further down, I get a sort of like a sample metadata table, which tells me more about these samples as to which ones are knocked down, which ones belong to a particular cell line, whether they are breast neoplasms or whether they are triple negative breast neoplasms. There are more information about the samples basically. So such an overview and such an infographic helps us to determine what essentially is this study, what is this study about, and would this study be a relevant study to be included in our analysis. I can also go back to the search results, and we can also take a look at another cohort. So this is the third data set, and again it takes me to a similar page where I have the information on the overall design and the number of samples, and when I scroll down, I get to see that how many percentage of the data set is a control, and whether what percentage of this data set is a breast neoplasm or a triple negative breast neoplasm. Here you can see that there are some which are of wild type, and there are some which are of knockdown. There are some genetic modifications that have been performed in this experiment, and you can get to see which, based on the sample metadata table, you can get to see which samples are of wild type, which are of knockdown. And this is essentially very important when you are trying to select the dataset for your analysis. So this makes it extremely intuitive and easy to compare datasets and to decide which one to include in your dataset, in your analysis. Another feature, which is one of my favorite features of this platform, is to be able to visually look at a cohort and perform some analysis on this cohort. So here I'm going to show you how we do that. So I'm going to select the dataset of 24 samples. And when you click, there is a button called options here. So when you click on options, there is an option to analyze the dataset. So when you click on analyze, it will allow you to analyze this dataset using other tools or utilities that are available on their platform. And one of their application called Phantasus is a web application for visual exploration of the dataset. So I'm just going to visually explore this dataset. So it takes a couple of seconds to load in this application. Once the dataset is loaded in Phantasus, you can sort of see the entire cohort here, and here you'll see like a sort of a heat map which provides you with the gene expression values for all the genes in that dataset. So these are the normalized expression values, and in addition to the expression values, you'll also be able to see the metadata and the additional data that is available for this cohort, and these are interactive, so you can just hover over these annotation bars and it will tell you what group a particular sample belongs to. And this is a very powerful method in terms of visualizing the expression of your gene of interest in a particular cohort, because it's just very intuitive. It's just point and click. And if you're interested in, let's say, in expression of a particular gene, you can just use these search bars where you can search for gene name. And here, let's say I'm just going to enter a name, BRCA1 gene. I can directly get the expression of that gene across the entire cohort. I can see what the expression looks like. I can also get additional information about this gene in terms of the location of the gene, the karyotype band, also whether it's a protein coding gene or not. And it can be helpful to quickly look up the expression of the genes in these cohorts, as well as compare the expression of genes across this cohort. This platform also allows to perform some basic preliminary analysis like differential gene expression analysis. So you can go to the tools, and here you can go to the differential expression drop down, and you can choose lemma. And since I know that this dataset has some samples as treated and some as untreated, I'm going to go to the description. And here I'm going to choose one group as treated cells and the other group as untreated cells. And I'm going to click on OK. So right now it's computing the differential expression log fold changes for each gene in the entire cohort. And once it finishes performing differential expression, you will see additional columns here in terms of log fold changes, average expression, the t-statistic p-values associated with, and the adjusted p-values. So this makes the exploration of a cohort extremely intuitive. So you can again go to the gene of your interest and go to that row and see what is the log fold change expression in that cohort, which can give you an idea on whether the gene that of your interest is differentially expressed in this cohort. So this is a very powerful method of quickly exploring the dataset of your interest in terms of looking at the expression of the genes, comparing the expression of the genes, performing differential expression, and also performing some additional basic preliminary analysis with your cohort. There are additional utilities that you can use in addition to performing differential expression analysis. You can perform clustering, you can even generate plots, you can perform a PCA plot, you can perform a volcano plot, you can perform pathway analysis, you can also perform a lot of other things. You can also annotate your rows and columns. So there are a bunch of different things that you can do with this web application, which allows you to perform some basic preliminary analysis as well as to visualize your cohort in such an intuitive manner. And lastly, talking about downloading the data. So to download a data set of your interest, you can move the data. You can click on move data button. And when you do that, essentially it will perform some sort of backend processes. The backend processes will run, and you will eventually have to go back to your OmixAtlases. And when you go back to the OmixAtlases page, the demo bulk RNA OmixAtlas is where you will be able to see your moved dataset. So when you click on view all data, I had previously downloaded or moved other datasets as well. But essentially this is the dataset that I moved some time ago. So now the ingestion has been completed. You will be given a notification that the ingestion has been completed, and you will see that your dataset of your interest has been moved here. And now when you click on options, you will be able to download, and the format that you will get your dataset is the .gct format. Basically, a .gct format is tab delimited text file which stores the gene expression data, and basically you can use this data to read in R, or you can use this data to read into additional programs and perform the analysis that you wish to perform. So that brings me to the top 5 features of the bulk OmixAtlas on the Poly platform. So the first is a sample overview that is provided for each dataset on the platform, which includes the dataset feature, the sample metadata, and the informative charts, which allow us to explore the cohort further. The second feature that I really like are the filters, which helps to make the search very specific. The third feature is the ability to use the logical operators as well as keywords to make the query and the search terms very specific, which will result in very specific and relevant search results. And another feature that I really like about the Poly is Phantasus, which is a web interface or a web tool which allows us for the visual exploration of the cohort. And in addition to visual exploration, we can also perform some basic preliminary analysis in order to look for the genes, the expression of a genes of interest, or also to look at differential expression analysis, or to perform basic visualizations with the cohort of our interest. And lastly, Poly also provides customized curation and processing of the dataset. In addition to these features, there is also one additional feature that I haven't explored today, which is a programmatic interface called PolyPython, which allows users to perform SQL-based querying at datasets, at samples, or at the feature or the gene level. So it also allows users to create cohorts, perform basic statistical analysis, and visualizations. So that's all I had for today's video. I would like to thank Elucidata for collaborating with me in this video and giving me the opportunity to explore their bulk OmixAtlas and talk more about it in today's video. I will come up with a new video hopefully soon on my channel. So please stay tuned and please subscribe to my channel, like the videos and share it, and leave your comments under the comment section. Until next time, see you.